Warren H. Wagner. And you will see that that makes complete sense in terms of the symposium. Tryon was a biogeographer. We've cited several of his works today in biogeography and talked about his centers. That was a very heavy influence on my thinking. And so as I developed as a graduate student, I was heavily oriented to biogeography. However, I went to what in those days was considered to be the dark side by consorting with the enemy, Herb Wagner, who turned out to be just a fabulous influence on me as a scholar. And he, as I think many of you know, was a principal architect of our understanding of allopolyploidy and speciation in North America. And so I'm sort of a merger of the Wagner and Tryon schools in many ways, which was just a delight to sort of bring the two together in my work. What I am eager to do today is to give you a sense of my present situation in terms of some of the work that's been going on in our group for decades, just to give a little insight, and then to end up with a sense of the situation as I see it, and I'll take you through it. And I'm going to do this by telling some stories, and what I've done is to manipulate myself to avoid all the stories that have been told in the last 2 days, because there's been a lot of biogeography and what are we talking about? Reticulation. There's been a lot of biogeography and reticulation in all of the talks, not just in this symposium, but also in the great set of Fern talks we had yesterday morning. We can fit them all together into one grand portrait of our situation as a bunch of scholars interested in the subject. But I have chosen to attack this in a way that I've done before, and that is to think about the possible time since the origin of these polyploids, and organize my thinking. I have sort of a predilection for this idea, which is a Ledyard Stebbins idea. It's nothing new. It's 60 or 70 years old now, since he first published the idea that polyploids display their diploid progenitors as they increase in size. So I will begin with my favorite recent origin story, and it involves two scholars who have been in our group, and this goes back 4 decades, believe it or not, or at least 3, I forget, I've lost count. And it begins with work that Morgan has always talked about with Kathy's resolution of there being an allotechiploid in Vermont, Verde montanum, which you've heard a lot about, so I don't need to tell you a whole lot about it. But where I'm going with this is to expand what Morgan talked about to some thoughts about the biogeography of this complex. And we've gradually gotten more and more of an understanding of what's going on there. And what Kathy revealed was that we had 2 ancestors, which have already been mentioned, and their global distribution is quite interesting. The luticum, the serpentine lover in the east, is present in the west as a serpentine and non-serpentine plant, makes it all across the west and out into the Aleutians, which is a suspicious distribution for anybody who knows Asian-North American relationships. It suggests you better damn well look in Asia for some solution to this problem. And so if we look to Asia, it gets even more interesting because the potatum is there as well. Again, this is Kathy's data, and the near allies to potatum and the others in this group are in the what I call the Sino-Himalayan region, and this is why I brought this thing up with Jacob, because I really see a difference between what's going on in the east of Asia and what's going on in the Hangtuan and in the Himalayas. And one thing that you pick up from looking at the distribution of these plants is that there's a sort of North Chinese to Hangtuan distribution piece, and there's more of a insular Kamchatka distribution of these plants. And this, by the way, is obsolete because it doesn't show a luticum in the Cascade, which you now have? Yeah. Maybe Kathy had it too. I frankly don't remember. Jinmei Liu showed up and worked on the Asian part of this story by looking at what was going on with the Asian plants in the context of a much larger analysis. And what was interesting about this is that she was able to resolve the relationships of the Asian taxa, with the East Asian, the sort of Sino-Japanese group being the earliest divergent, and then leading to a dichotomy that leaves behind the Eastern North American pedatums, and then brings together the Chinese, the North Chinese Hangzhuang plants with Aludicum, which is again that connection between the Hangzhuang and what's going on in the Aleutians, which we have in other parts, as in Polystichum as well. That gave us some great insight into what was going on with these plants that we had not had before. So that gives you just a little flavor of the, I'm blowing through these things. We just got a little taste of what the biogeography has been like of these reticulation events. And what's key here is that you can see I've got it calibrated to time here in another diagram from this paper. Jen May actually did a piece analysis and got the timing. And the thing that stands out here is these events are all really recent. These are late Miocene to Pleistocene. So we're talking about recent events. And of course, superimposed on that is the origin of Viridium montanum, which, and this shows to be Pleistocene for sure. Moving on, I want to talk a little bit about one of the most recent projects in our lab. This is talking about the origin of Polystichum californicum. There are several people in this room that I've shared these ideas with, and they've been kind enough to be excited about it and also provide plants, which has been wonderful, especially Carl Rothfeld, who actually contributed all the cool, confusing plants. So in 1973, Herb, this is one of his classic projects, demonstrated that californicum was an allotetraploid and it was derived from a diploid, Dudleyi, and at that time what we called Munitum in the broad sense. We'll complicate that in a minute. And these plants are all in this one picture by John Game. You've got the diploid here, you've got, the twice pinnate species Dudleyi here, Munitum sensulato up at the top, and there's a couple of fronds of californicum peeking in from the side with intermediate dissections, so that picks up nicely. The key thing that Herb figured out was that they're actually both sterile diploid and fertile tetraploid versions of californicum. And Wes will say, well, why haven't you named those two? But I haven't gotten there yet, and not sure I want to. But the thing that stands out about this is it means that this is a nascent polyploid complex, that there are still sterile diploids right alongside the tetraploids. And Alan Smith developed and supported that by finding sterile diploids in a completely different part of the range, farther south in California. So this is a young lineage to go with the Adianum pedatum, the Viridium montanum origin lineage. Now, along comes Melita Schmeck-Pepper, who was one of the long line of students that I had. She's an old professor who, when I first got to UVM, said, Dave, whatever you do is surround yourself with your intellectual superiors. I got to tell you, I've done that in spades. I've done it over and over again, and they make me look good. You might think I'm as smart as all these people, but that's not the way it works. It's been a great privilege to have all of you who've worked with me in this room with me. It's been a joy. Melita was one of these folks, and she combined an amazing set of data sets. There was chloroplast DNA data, there was nuclear DNA data, there was flow cytometry, there was morphometrics, and I added spore measurements to that. So this is a fully integrated data set. There are a couple of cool things that came out of this. Dave Wagner, who's sitting with us today, came up with this basic idea that the Munitum idea was actually three entities, two species, one of which had two subspecies. And Melita's work beautifully confirmed Dave's taxonomy, because the species imbricans came in two flavors, imbricans, subspecies imbricans, subspecies curtum, and they were sister to one another, and in turn sister to Munitum. So fully supports the taxonomy. The other thing that was really interesting right off the bat, that even though there are 3 entities in the complex. All of the Californican entities that we sampled, the tetraploids that we sampled, and almost all of the diploids were derived from curdum interacting with Dudleyi. So although there are 3 candidates, there's only one that's really prominent in the history of California, right? And that was exciting geographically and ecologically. Just to give you a sense of how bad it got. As I said, a lot of these plants are ones that Carl got lucky and put together in his sample. But here's the here are the 2 Californicum variants, right? We'll call this one Curdum cross Dudleyi, just to be keep from naming Californicum, and the 2 parents. And then we have backcross in the data set. Dudleyi cross back to Californicum crossing back to Dudleyi was there, and that's rather ugly because it's hard to tell. It's barely different in dissection, but a little bit. And then we actually found Munitum sensu stricto in a sterile diploid. So we did find one of the other possibilities in this data set. And the most awkward thing was that this, we obviously couldn't do DNA on this thing. It's 140 years old, although I suppose it's possible. We've done that for other groups. This is the type of Californicum. It has 2 features that are awkward. It has abortive spores. So it's either a sterile diploid, some sterile entity, some hybrid. And it's got a morphology that's intermediate between Californicum and Unitum. So tentatively, this thing is actually the Californicum Unitum backcross. So we have a name problem there. The thing that's really interesting ecologically and geographically, and this picks up on some of the ideas we've had a rich set of ideas about ecology and geography today. If we look at Dudleyi, it is confined to these redwood groves near San Francisco and points south. Really cool, great country, high moisture habitats. And we look at Curdum, it goes all the way to the Mexican border, extends all the way up to the northern border of California. In our sample, we actually we discovered a plant from Central Oregon, so even further. And these are generally in drier habitats, different conifer species, more open habitats, sometimes extremely open habitats. And you might think of it as the most divergent from Dudleyi in its ecology. And you will see that Californicum is an amazingly broadly distributed plant. So this is a great example of a tetraploid that's expanding beyond the range of at least one of its progenitors, and probably both. And I just wonder if, and so that we can pursue in the future with ecological analysis, that combining these disparate genomes gives the niche breadth to the tetraploid that's allowed its success in this case. I don't like that thing. Fourth thing I want to talk about is Stacy's master's project, which is Polystichum brownii. And this now we move to what I call an older polyploid. And in summary, we're going to see that its origin is calibrated to have shown up earlier. And the other thing that stands out about this plant is, yes, it's an allotetraploid. We have no idea what the progenitors were. They're either missing or extinct. And people have been looking for this, for the ancestors of this thing for decades. Now, talk like this would not be complete without mentioning the grand old lady, Irene Mann, who in 1950 published the book that we might as well call the Bible of reticulation studies in the ferns. And she, in that book, demonstrated that brownii was an allotetraploid, and her student, Anne Sleep, who was just a spectacularly successful teratologist and died too young, demonstrated that none of the European species could be progenitor of this, and went on to show that a number of the North American species couldn't be progenitors. Stacy's work took samples from throughout the circumboreal lineage of brownii. We were lucky enough to get the samples from essentially throughout the world, through most of the endemic areas across the circumboreal range. and put it together to discover what the heck was going on. So the thing that stands out about this is that if we look at, in this case, look at the nuclear data set, this is actually, now we have a combined pair of nuclear loci. It's a very nice piece of work in that sense, as in many senses. I don't mean to imply it's the only sense. And we discovered that we have two clades that are prominent here, and they both contain Brownii clones. But this clade has as its sister Lemoonii, a North American species. And this clade is nested amongst a set of both Hengduan and Sino-Japanese species. So we have this peculiar situation where the genomes in this plant have relatives in totally different hemispheres. So how to resolve this? This is actually something that can be imaginable, and that's the great biogeographic question. Could these things have arisen from some sort of meeting? And there is, in fact, a meeting place for these characters. It's Beringia, and there's a lot of literature on the idea of Beringia being a meeting place. But I'll just share with you a couple of images of Beringia being above water, which it has been not only in the Pleistocene, but earlier on, well back into the Cenozoic, including into the early Miocene, which is the calibrated historical origin time for Brownii. Now, one of the cool things is we have a lot of fossil records of angiosperm flora from Beringia from that period of time. And so we're able to decide whether the angiosperm flora that was present in that, at that period of time matches the current kind of vegetation we expect to find Brownii in. And I actually did a Maxent analysis on this and went through the whole 9 yards. But the short answer is yes, that the conditions in Beringia in the early Miocene were appropriate for the first Brownii plants were they to be generated there. So wild and crazy, maybe, but it's a hypothesis to be disproven that there was a meeting ground in Beringia in the Miocene where these two genomes met. The fourth thing I want to talk about is a little bit of, and now we're getting to really old, is to talk about Australopithecus. So this is a great group, and I was fortunate enough to have an Argentine scholar express interest in coming to our lab to work on the Austral South American Pithecus. So I wanted to share with you some of her work. And just to give you a little flavor for this, it's just a classic disjunct austral distribution for this genus. You can see that there are Southeast Australian plants, there are New Zealand plants, and there are Southern South American plants. And at slightly more northern latitudes, there are plants in Brazil and in Southern South Africa as well. So as I said, Rita joined us, and she joined us to do molecular work and developed chloroplast analysis. So this, she was also an accomplished cytologist. So she actually did chromosome counts for all these plants. And she was able to integrate the chromosome work and the chloroplast DNA work in thinking about these plants. So first of all, what she found was that all of these austral lineages were part of a monophyletic group. And in particular reference to the Australia, New Zealand, and Southern South American groups, they are a single monophyletic grade, and then clade. And then within that lineage is the Southern South American group, which is again a monophyletic lineage, suggesting that the history of these things is that they arrived in South America from points west. Now, the thing about this is that this whole set of events is, well, it's mid-Miocene, 13, 14 million years ago. So it's a while ago, and you might say, that's not that long. What are you talking about, ancient polyploidy here? Well, just a minute. But the point is that's recent enough so it has nothing to do with vicarious biogeography. It's not about these plants having been on Gondwana when then split. It's about long distance dispersal, in this case west-east into South America. And that is the trend in all of the analyses across both kingdoms in the monoliterature. We're just teaming up with the rest of the people who are discovering the same sort of pattern. Long distance dispersal in these very commonly shared floras in the far south. But the thing that is really fascinating about this is that here at Rita's chromosome counts, she got counts for all her plants, and the plants from Australia and New Zealand have also been counted. There are no diploids anywhere in this column. So yes, there are polyploids, and the neopolyploids are off the board. So this is then a history that is superimposed on an earlier episode of Origin of Polyploids, for which all the diploids are either missing or extinct. And it'd be kind of hard for all, we're missing, I think there's some missing diploids in here. And so I think what's exciting about this is we're seeing that deep history in which the tetraploids are taking over from the diploids. There's another group, a completely separate group in a splenium worked up by the Australians, in which you had seen the exact same pattern. The polyploids are all octoploids, and the basic species are all tetraploids in the lineage. So there's something going on in the south that we need to think about. In other words, what is it about the two hemispheres which are so different geographically that means that the ancient polyploid is represented by these lineages. The last thing I want to do in the way of lineages is to talk once more about Nikki's work. This is the one that she didn't talk about today, and it's the last chapter of the PhD thesis. And the reason I want to talk about it is it's complicating things by throwing apomixis into the mix, right? And Amanda did a great job of setting us up for that, and Nikki threw it in as well. So we've had a good flavor of that, but I want to just articulate the same set of ideas for a different group. And it's our local Northeast North American tetraploid apomix. Now in press is being named Phegopterus excelsior. And this work is work of Nikki in collusion with Art Gilman and Susan Fawcett, both of whom have talked here today. And this was built on earlier work of another one of my students, Heather Driscoll, who worked with isozymes and morphology with Art and me earlier on. And this tetraploid apomix then is just local from Connecticut into the Maritimes up into Ontario. First discovered by the Canadians in the 1970s. And just to give you a little context, for a small genus, this is an ugly little thing. It's got an awful lot of complexity to it. There are only 4 species, maybe 5 known. Here's the one you may well know if you're into ferns. It's the triploid apomix, Polystichum connectilis, the Long Beach fern, all over the northern hemisphere, one of our circumboreals, right? There is a diploid sexual race of this in Japan, which we had access to, thanks to Matsumoto, who's the scholar who's really worked on this group. Here's the one we've already talked about. Here's Excelsior, the Eastern North American apomix that's tetraploid. And then we have another sexual diploid, and that's Hexagonoptera here in North America, the broad beach fern. And then we have the morphologically disparate Phegopterus decursivi pineda, which is just East Asian, and it's the bad boy in the mix. Diploid and tetraploid sexual lineages, a triploid apomix, and outside the realm of what we did in this project. So this is this elegant data set. At least I think it's elegant. I still have to convince Nikki that it's elegant. But I just want to give you a little sense of the territory. So what we've done here is to show nuclear data. This is for ITS, to try to get a sense of the distribution of genomes, but overlaid on what we know about the cytology. Fortunately, in this case, we know all the ploides for all these plants. I've already talked to you about that. And so what's interesting is if we look at Connectilis, there's only one genome in it. Okay, so that suggests that it's an autotriploid, which I've indicated here by calling it CCC. Right? There's and that genome is perhaps originates with the diploid Connectilis. So there's nothing inconsistent with that hypothesis in the data we've got here. Hexagonoptera, decursivi pineda seem to have nothing to do with this history. Genomes that are found in these plants are not found in any of the others that we're working on. Right? But if we now ask about the tetraploid, There are two different genomes in the tetraploid, Apomictic excelsior. One of them is sister to the triploid connectillus, right? And the other is a mystery genome. Right? So it looks like what's going on here is that triploid connectillus has contributed to this tetraploid, which is comfortable because the two grow in the same woods. Okay, but this other character, which would be the fourth genome, would most probably be contributed by a missing diploid progenitor. And Susan, you'll notice I've come over to the dark side. Susan won this argument. It was all a question of parsimony, and I think she won. But in any case, it looks like that some missing diploid progenitor contributed the last genome to the tetraploid. Now, what's up with the geography? And we have to convince this thing to go forward. There we go. What seems to be, this gives you a little sense of the distribution of these characters in the world. We've got the weirdos that have nothing to do with the story in red and purple, one in North America, one in Asia, and one in North America. And then we have the widespread connectillus in yellow, and then we have our local tetraploid apomix here in Eastern North America. So on a global perspective, the most likely origin is that there's a missing diploid progenitor sitting under our nose in northeastern North America, and we better get back in the woods. I've heard that somewhere before. However, traditional mountain apomictic dogma is that apomix contribute sperm, because in many cases we see the gametophytes in the dirt mountain apomix not having functional archiconium, but making good sperm. So it's common in the literature to argue that they will be male parents. But so for this hypothesis to work, Phegopteris connectillus has to be the egg parent in this history, which is a rare event for this kind of apomixis. Right. And there are two apomictic pathways. There's a second, the Braithwaite pathway ordinarily has, or commonly has, female contributions to offspring. Well, that's my set of five histories. To give you a sense, so 4 histories which give you a sense of my feeling about history and the how long ago these events take place, which I find fascinating. And the 5th one, once again, sort of facing up to the challenge of apomixis in these stories. So what I want to do, before I do that, I want to go here. I decided to do this in a different order. This is a cartoon that attempts to, it's actually something that Amanda said, and that I think other people have said today. And that is, if we're going to do this reticulation business, it is very wise for all of us to be thinking about hybridization, apomixis, and polyploidy at the same time. And what I've done here is attempt to put some of the possible pathways into one diagram. So we have in any case, we have some divergence in the history. We've got to have divergence to have some hybridization. But there doesn't have to be hybridization in these histories. We can go directly to autopolyploids, and we can go directly to apomixis, as we just saw hypothesized for Phegopteris connectillus. But of course, at the same time, we have a lot of triploid apomix, and a number of these have been demonstrated to be allotetraploids. And then, of course, we have the classic allotetraploid sexual races. Well, any of you who have done this work know that you start filling in lines, and it just goes crazy. And I thought I'd just, share 3 of these with you. 2 of them you've heard people talk about today, the Myriopteris yavapensis story. Then Amanda told us the story about the Xiphopolisticums, including Susumensi, that Nikki told. And if you really want to go nuts, look at Hori's work on Dryopteris varia, which is even worse. So we are up against some serious ugliness here, folks, and it will pay for us to constantly be taking every possible tack we can to these. And I want to take you back to what I'm now calling my game board. Okay, and this is where I want to end up with you. And think about this, is how do we represent what we do to naive students that show up in our labs, right? And I actually had a naive student show up in my lab this summer, and I said, another victim. And I realized that I didn't have like the big picture to give to the student. So I proceeded to try to put it together. And this is what this board is trying to represent is the sources of information across the life cycle, right? So what I'm calling life stages, spores, gametophytes, and sporophytes. And then going down, the kinds of information that we have been fortunate enough to apply to these. And my argument is, look at all the stages, look at all the sources of data. It's not, it's not just what a lot of us, and including me, are really happy doing these days, the DNA. These sources are all powerful sources, right? So just to give you a little sense of it, what I did was to try to emphasize the sources that have been used in this symposium. And I may not have gotten them all, but that's what this is. So in the talks in the last 2 days, yesterday morning and this afternoon, this is the sort of my idea of how far across the game board we have gone in looking at these stories. And it just says we're in a great place, that we are looking at these different data sets and we're integrating them into powerful stories. And the great thing about these data sets is that they're mutually informative, that they allow tests of hypotheses. And you don't necessarily start with DNA. You may start with a board of spores. Or, the great paper yesterday morning where she was just counting or looking for, in the apomix, counting the number of spores per sporangium. That's right there. You can start there and you're making real progress. So we have a number of different ways to think about going about this. And real powerful tools. Tools that I cannot imagine us even dreaming of when I got into this business 45 years ago. And that leads me then to just come up with a few closing thoughts. And the first one is the power of integrating the contributions, of thinking about the contributions of hybridization, polyploidy, and apomixis as we work on reticulation. And so many people now have this in mind. It's terrific. And then the ecological dimension that's really emerging from especially work like Morgan's, but also the work yesterday on gymnocarpium, in which we look at maxim analyses and project those climate distributions back in time. That seems to be based on inferences about climatic contributions to niche. And that stuff is going to be, and I realize now what I've done is talked about number 4. I got here too soon. So this is what I'm saying. We want to consider biogeography in the light of climate variation, both across geological time, but as some people have started to work on, especially Michael, what's going on with elevation variation in terms of climate over the history of these things. And that has come out especially in thinking about the Costa Rican and Andean work that we're on. And then I touched on the idea of developing our sense of niche and distribution. They're intimately connected, as you've seen. And there's a little bit of thinking today from Li Yuang about so the whole idea of how the niche of the polyploid is related to its progenitors, but it does seem like they're broader in general. There's many cases in which that's true. And yes, it takes time for the polyploids to occupy their potential niche, but they're really good at long-distance dispersal, among other things. We've got a lot of proof now that tetraploids and other polyploids tend to be selfers, so they're better dispersers just on that account, and perhaps in other ways. So this is where I think we're at, and I just want to say it is a privilege to be part of this community, exploring all these ideas, and we hope to be around a long time watching these things happen. Thanks a lot.